we, with basic tests in the lab, say that, oh, this particular compound is showing good response. That is, it is inhibiting, if it's an enzyme, inhibiting the functioning of that enzyme. Okay, so by identifying that, now remember the target itself is, you know, a bigger molecule. It could be an enzyme, a protein. So surface, or that pocket receptor. And then the third area where chemists really get involved is in what we call the heat to lead optimization. So, like I said, the first, we identify the compound that is showing some level of response. We call it a heat compound. And then we go on to, in most cases, the heat compound, for instance, may not be soluble. Okay, it's active, but you cannot get it into the body system. It's only maybe water soluble, will not dissolve or enter into a fat-based system. Okay, so we then have to, through a series of careful steps, try to understand how to move that compound from just being a heat to promote all of the chemical properties of interest such that it becomes a lead. Okay? So a lead compound is the kind of compound we are interested in, that we actually move into the stage of drug development. Now, so you have your heat. The challenge is that when we generate those heats and we evaluate them, at times some of them fail, and so the process stops there. Okay? Even after you have identified the heat, while you are validating the heat, unfortunately, some of them also fail and the process stops. So what I mean is that we can start with, once we have identified a heat compound, we have ways of doing that. We generate its derivatives, compounds that are similar to it, and then we try them. But unfortunately, some of them will fail. Okay? And when they fail, we just put that aside. We continue with only the one that passes. So for a compound to go all through, it must go through each of these stages to the point of saying that it has actually been optimized as a lead compound. Now, it is only lead optimized compounds that actually are considered as potential drug candidates. And so, on the next slide, you will see that all of what we do in basic research, okay, which takes a lot of time. In fact, you could go to the point of having so many compounds, more than 5,000 compounds, and in some cases, none will make it through the process. So when I say it takes time to make a drug, now that is the angle we are coming from. It takes a lot of time to actually be able to get a compound that makes it through the entire process. It is after it has gone through, okay, as, you know, a lead compound, it comes out, we say, okay, we're on a drug discovery path, then it enters what we call preclinical trials. Okay? And at that stage, we start looking at the kind of effect it has on, not man now, but animals that are considered, you know, to have a similar system by which we can infer. So if you test it on mice or on rats and it kills all of them, you know that it must not, that one has failed, right? So even though it has made it through all of the previous stages, if it gets to that stage and it's, you know, causing trouble, then we know that is out. Okay? Professor Nia is a toxicologist, so I'm sure he too can give us some understanding. So basically, the compound must go through all of these phases to be able to actually get to what we call a drug that is approved to be on the market. The slide below, sorry, still on that same slide, the image below now basically tries to help us summarize that if you start with 10,000 molecules, in many cases you end up with one approved drug. Now, so that is to give you an idea of how expensive the process actually is. And then look at the length of time there. It says 12 to 14 years. Now, there are compounds that you know that you take today. I can tell you that it took more than 14 years to actually make it out. Okay? But obviously, when there is a breakthrough and you now have a drug, it then is not so difficult to make other, you know, if there's chloroquine, you can make camoquine, and you can make, you understand? So that's not, what I'm saying is to get the one drug that first of all is able to attack the disease in question. All right, so, and the process, as you can see, would vary depending on where the research is being done, but approximately $2 billion US will be involved in that entire process. So it's from preclinical drug development to clinical drug development, where you have phase 1, 2, and 3 trials, and then to market drugs, which is now being described as phase 4, actually, after that process. Because it can go through everything, and then you realize that when people take it, it has a different effect. We have tested on animals, it is fine. We have tested in the lab, it is fine. But when humans take it, and that's why you see that in those slips in the drugs, they write things like the kind of side effects you should expect. Anything beyond those side effects means there's a problem. Okay? So they keep monitoring drugs even way after it has been released, after it is now on the market. You still have to keep monitoring. Because, like the guy said earlier, okay, everything is chemistry. Okay? As I'm talking, there's a flow of ions. And you can hear me audibly because as I speak, chemistry is involved. So if the particular body system is somehow not responding well to the chemical variant that has been introduced, it may work for others. But the reason why some people don't use the same drug for malaria as other people is because it doesn't work for them. Okay? So what works for one person may not work for another person. And so what will give a reaction in person A may actually be fine in person B. All right, so let's go on to modern innovations in drug development because of our time. Now, the modern era, of course, we have so many innovations today. And we know that with time, we have actually advanced significantly. So one of the things that is an area of innovation is the fact that we can today have what we call targeted drug design. Okay, and what we mean by this is that it is actually possible to move away from broad spectrum drugs. If you followed the presentation of Professor Nia very well, he actually alluded to this, that it is possible to have more targeted types of treatment, okay, that is literally specific. It may not be just person specific, it may be specific to a class of people, all right? For instance, paracetamol works well for some, but some people need to use aspirin, okay? I don't know if you ever looked at the history of aspirin and why aspirin was formed, but I won't go there now. So, this basically is because today we can do what we call structure-based drug design or fragment-based drug design. Okay? And by these techniques, we are able to actually go specific with particular biological targets, which in short means we are able to minimize the off-target effects and improve the drug efficiency. Okay, so the first one is targeted drug design. The next one perhaps is a word we are more conversant with, nanotechnology. also applied in drug delivery. So it is possible to use what we call nano-sized drug carriers, okay, such as liposomes, and use them in such a way that first they protect the drug before it gets to the site where it is needed. Okay? In fact, today we administer some compounds. Some of the drugs you even buy now, you are buying a form of the drug. It is when it gets inside you that it metabolizes into the form that actually works. Pro-drugs. So you have cases where we can also embed the drug within some kind of capsulate form that allows the drug to transmit and get to the site of action before it actually delivers. So this is all about, precision targeting for drug therapy. The next is chemical biology and bioconjugation, which simply is about biologics such as antibodies, and these have been used. Now, so this may seem like it is more biology oriented, but remember, at the end of the day, even H2O is still a chemical. All right. So, but essentially, we have been able to develop what we call antibody drug candidates. Okay, and these are able to deliver to specific cancer cells. And also, we have controlled release systems. In this particular case, we use polymeric materials, okay, to administer a drug. But the point is, they are able to protect before it is have been able to develop what we call antibody drug candidates. Okay, and these are able to deliver to specific sites, for instance, cancer cells. And also, we have controlled release systems. In this particular case, we use polymeric materials, okay, to administer a drug. But the point is, they are able to protect before it is time for release, and also able to release in what looks like a dose-dependent fashion. All right, so the role of chemical innovation in clinical trials. Thank you. Help in both in vitro and in vivo models, okay, to predict how new drugs will in a person. Of course, biomarkers have also been able to help us, okay, in this kind of monitoring. Also, pharmacokinetics and pharmacodynamics, which are very important parameters we look at, especially in the preclinical stages. We want to be able to measure precisely the level of drug absorption, the level of drug distribution, the level of drug metabolism, and drug excretion. Now, these are very critical parameters for any drug, because you don't want to use a drug that is just accumulating in your body. You don't want to use a drug that is not being distributed properly and you can get it out as at when due. You don't want to use a drug that isn't absorbed through the tissue. You don't want to use a drug that is just accumulating in your body. You don't want to use a drug that is not being distributed properly and you can as at when due. You don't want to use a drug that isn't absorbed through the tissues of the body. Okay, so mass spectrometry and bioassays have also proved very important in this regard. Now, from laboratory to patient, just taking a look to actually determine very clear standards, okay, for drug approval, because a drug needs to be safe and effective. So, when we say cure for cancer, for instance, they are still struggling with cure for cancer. That's not really the correct way to put that statement. There are cures for cancer. The problem is we need a cure for cancer that is selective for cancer cells. Okay? If it cures cancer and if it kills cancer and kills normal cells, it's not really safe. Okay? So it's not that we don't know what can kill cancer. What we need is selectivity in cancer treatment. Okay? So these are things that we can obviously, with our knowledge of chemistry, advance today. And of course, personalized medicine. Like I said, the keynote speaker already alluded to this. You can have ways of using pharmacogenomics, to adjust dosages and select the most effective drug for a particular. Of using pharmacogenomics, to adjust dosages and select the most effective drug for a particular individual. And lastly, on this post-marketing surveillance, okay, after the drug is already out, we keep monitoring. Okay, if there are things to check, if there are side effects that were unexpected, they need to be checked. The classical story will be the case of thalidomide. I don't know how many of us remember the story of thalidomide. As we begin to round up, the impact on patient care, of course, is key. I mean, what we are looking at is how all of this affects the care for the patient. Transformative therapies, as a result of chemical innovations, have been born, and breakthroughs, particularly in the area of immunotherapy, precision medicine, and gene editing, of life. Okay, new formulations, intravenous treatments, all enhance, the patient quality of life. And lastly, on this, reducing healthcare costs. So while drug development, of costs, of course, we know is expensive, we know that we can afford to reduce the long-term healthcare cost by producing drugs that are more effective. And so the last major section will be current trends in chemical innovation, artificial intelligence and machine learning. AI is not just the next buzzword. AI is truly something that has come to stay. So artificial intelligence is also involved even in the area of drug design. Okay? Because of our time, I think I'll just leave it at that. Next is green chemistry. Green chemistry is looking at how we can remain environmentally friendly. We're not just trying to make drugs at all costs, even if we are killing the environment. We are trying to look at eco-friendly to reduce waste in the environment. Gene editing, okay, which again leans more to the biology side of things, is also very, very critical. And lastly is drug repurposing. I should say something about drug repurposing. Now, you see, there are compounds that have made it through the entire clinical trials, but ended up failing when we actually use them to treat the actual infection we wanted them to treat. Okay? But if you remember during the COVID pandemic, while it takes all of those years to design a drug, you know that we started attacking COVID within 1 year. Okay? Now you should have been thinking that, okay, what exactly is this person saying if I said it takes 12 to 14 years to make a drug? But one of the things we did during the COVID pandemic was to look at drugs that are already on the market, that have made it through, and then begin to look at how we can repurpose some of them. Because we now have better understanding of the target, so we began to look at what we can use to hit that target. And so in repurposing of drugs, for instance, thalidomide, originally developed as a sedative, was later repurposed for leprosy. Okay? Metformin was initially developed for diabetes treatment, but it's now being explored even in cancer therapy. Okay, so this it just shows that at times we can save time by going back to look at the library. So there's no way we'll dump literature, where we'll dump all the things that have been done in the past. In conclusion, chemistry is fundamental to every stage of drug development. from discovery to delivery. Innovations in this field not only drive scientific progress, but also directly enhance patient care and make treatments more effective, personalized, and accessible. As we look to the future, continued investment in chemical research and innovation will be essential for developing the next generation of therapies, ultimately improving global health outcomes. By way of acknowledgement, I'd like to thank the organizers of this wonderful conference for inviting me. I'd like to appreciate the entire staff under the able leadership of the Dean for this opportunity. Thank you for listening.
Thank you so very much, sir. Now, the next on the agenda is the panel discussion, which is going to be moderated by Dr. Ogali. Please, let's celebrate her as she steps forward. Let's celebrate her. Are we tired? Let's celebrate her. Are we tired?
Good afternoon, ladies and gentlemen. This is a panel discussion session, so I'm not going to be doing it alone. So you kindly please join me, invite the following persons to come. Good afternoon, ladies and gentlemen. This is a panel discussion session, so I'm not going to be doing it alone. So you kindly please join me, invite the following persons to come join those on the high table so we can have Dr. Funmilayo Agbaje, please come up stage, and Dr. Mojisola Asowata, please come up stage. We're going to be discussing together the role of nutraceuticals in disease prevention. Professor Ayo Omoge is a professor. Professor Ayo Omoge, Helen, please come up stage. Dr. Funmilayo Agbaje, please come up stage. And Dr. Mojisola Asowata, please come up stage. We're going to be discussing together the role of nutraceuticals in disease prevention. Professor Ayo Omoge is a professor of food science. Dr. Agbaje is also a leading scholar in food science and technology. I am a phytomedic, a doctor of phytomedicine. And Dr. Asowata, please come this way. Yeah, can you help us with another microphone, please? So because the subject matter has to do with the role of nutraceutical in disease prevention, the first thing we want to do is to have all of our speakers tell us something about the concept of nutraceuticals. What is it about? I'm sure the students here are hearing for the first time, so can you kindly tell us what it's about?
Thank you very much. First of all, I'm Ayo Omoge Heli.
Hello.
Nutraceuticals are usually part of a food, or in some cases food, but basically they are usually isolated or presented in either isolated or concentrated form, and then their basic the basic concept is that apart from providing the basic nutrients in a food, in which case some of them meet basic nutritional requirements for humans, they are also used in the prevention of, and then the concept was actually based on the fact that materials serve as nutrient requirements. And in some cases, some of the food materials can also help in providing form of prevention for some diseases. And basically that's what the nutraceuticals are. And it's also important to know that they are not synthetic drugs. They are normally from natural source. They are sourced from natural sources like some of the food materials can also help in providing form of prevention for some diseases. And basically that's what the nutraceuticals are. And it's also important to know that they are not synthetic drugs. They are normally from natural source. They are
Functional food specialist. Can you speak to that?
Thank you very much, Dr. Ogale. Fumlayo Joy Gbenga Fabuso Agbaje by name, and I specialize on functional food chemistry. Then when you talk about nutraceutical, it's come from two words, nutrition and pharmaceutical. When you talk about nutrition, there are basic nutrients needed in the body, which could be in form of protein, crude fiber, minerals like calcium, magnesium. Fumlayo Joy Gbenga Fabuso Agbaje by name, and I specialize on functional food chemistry. Then when you talk about nutraceutical, it's come from two words, nutrition. They are bioactive ingredients obtained from natural products, like what Professor Omoge said, from food, plants, animal that have that can produce optimal health benefits for humankind. However, this nutrition from functional food, even though they are similar in some extent, functional food are the food products obtained from food components. Maybe you combine two or more food together, depending on their health benefit that they can have. For example, Gbenga Fabuso et al. Produce PGP biscuits from functional food that help to manage diabetes. So we produce biscuits from PGP and wheat flour. And we did the clinical trial and it was able to regulate blood sugar level. So we can call that one functional food, even though it's different from the nutraceutical. Thank you very much.
Okay, so thank you so much. You have something more to add.
Sorry, I also want to add that actually functional foods and nutraceuticals, they have some similarities. The first similarity is that they are from natural sources. They are naturally based. Secondly, they are actually meant to help overall health well-being. However, their differences, they are different in three forms. One is their format, the form of presentation, in which case functional foods are whole foods, like you eat your beans, your grains and the rest of them. But in the case of nutraceuticals, they are usually presented in form of tablets, in form of pills, in form of capsules, in form of liquids. So they are called dietary supplements. Another thing about the functional food is that the mode of consumption for functional food, they are normally not regulated. It's normal food that you eat. But in the case of nutraceuticals, they are regulated to be taken in as supplements. So if you are taking your food, normally you take your breakfast, your lunch, and your dinner. Nutraceuticals, they have to be taken at specific time and at specific dosage. So those are, and then their mechanism of action, they are usually different. Thank you.
Thank you, Prof. So, Dr. Osowata, as a phytomedic, I'd like you to address this issue from that aspect, but the question to you will be, these food scientists are arguing that nutraceuticals are components of food, combination of food, disease prevention, but I want to ask you as a phytomedic, is it okay for us to sideline or do away with traditional medicine, or let's not say traditional pharmaceuticals, let me bring it down, indigenous medicine like our agbo, because these are also naturals. They are claiming that nutraceuticals are naturals. Okay, so should we do away from those and just stick to nutraceuticals for prevention of disease?
Okay, thank you very much. My name is Abiola Mujisola Asowata Ayodele. As Dr. Gaya rightly introduced,
nutraceuticals, they are regulated to be taken in as supplements. Food, normally you take your breakfast, your lunch, and your dinner, But in the case of nutraceuticals, they have to be taken at specific time and at
Is that we should first of all look at what is that nutraceutical. That nutraceutical that we just mentioned here is about the nutrition aspect, that is what we take into our body daily, our daily requirements for our body. Then marrying it with pharmaceutical, that is what we require to treat our body. So when it marries each other, that is nutraceutical. That is taking something that will also help your well-being and at the same time managing an infection or disease. So when we now talk about nutraceutical, we are having different forms. That's, as Professor Omoge said, we have it in medicinal plants, we have it in dietary forms, we have it in functional food, we have it in so many ways. So, but doing away totally with our traditional medicine, that is not correct, and that cannot be. But the thing is that the particular active ingredients that is important in a particular plant is being extracted in more quantity.
Okay, maybe it's that place that you are.
Than when you take the plant alone or the food alone. For example, let me mention this Vitis and vinifera is in red wine. When you take it just like that, you take resveratrol in per liter at low milligram, converted to nutraceutical, it is in 500 milligram. So you take that, it addresses a particular chronic disease according to what you want to address is at higher concentration. Then we have lycopene from tomato, carrot. So anybody having an issue, probably you have eye defects or glaucoma or something, they will tell you take more of carrots because it consists the supplements, food carrot supplements that turns to nutraceutical consists more of that milligram in that nutraceutical than the one you just consume in the carrot. And there are several plants. We have agave, we have cyanobacteria. I have chapters on that. With that, we have, when we take the active ingredient in them to address the particular infection, it takes care of that infection at the same time our well-being. So I think
okay, thank you so much. Someone wants to add something? Okay, so that addresses it from the phytomedicine perspective. Traditional, our local preparations are not inferior to nutraceuticals. However, they work side by side, especially as it got to do with what it is that is the health condition. If it's a nutrition-based deficiency, then you should embrace all of nutraceutical. But if it's not a nutrition-based disease or infection, then you should want to think about traditional medicine, herbal medicine, pharmaceuticals, and the likes to manage the disease. Okay, moving on, Professor Omoge, you heard Professor Olomola talking about precision medicine, talking about how that a particular drug may not work for a particular person doesn't mean the drug is not effective. Does that also affect the efficacy of nutraceuticals? Is person to person difference, genomic makeup, or does it affect the efficacy of nutraceuticals? Please speak to that.
Thank you very much. Usually, even though nutraceuticals have benefits, they are also limited. Everything in life has its own limitation. We may not call it disadvantages, but of course they are limited. One of the things is genetic differences or variation. Some people, as a result of their genes or genome, they react differently to particular nutraceuticals. I heard of a story some time ago. It actually happened, not like a story. The person who was there said it. You know, these people that sell agbo, our local women, you know, she just carried it, brought it by, and a cluster of men were seated, and others got the same quantity and drank. And one particular person also drank the same quantity, and the person stretched immediately, and he died. Within seconds, he was already gone. So, not
deficiency, then you should embrace all of nutraceutical. But if it's not a nutrition-based disease or infection, then you should want to think about traditional medicine, herbal medicine, pharmaceuticals, and the likes to manage the disease. Okay, moving on, Professor Omoge, you heard
negatively. All right, so it is important that before people go on to take these nutraceuticals, they should check their genomic makeup, their resistance to drug, their adverse reactions. Some people have allergic reactions. There are things that are not drugs, but I don't take personally. For example, I don't take honey. Not because it's poisonous, I don't try it. Every time I have tried taking honey, I ended up in the hospital. All right, because my system reacts negatively and differently. So you should be able to check. So as a result of genetic makeup, it can affect your acceptance or the efficacy of the nutraceutical. So genetic makeup and then microbiome difference too can also affect the efficacy of the nutraceutical. Thank you.
Okay, I can see Dr. Agbaje wants to react. So I'll just add another question to you. She has already given you a pointer that there's a problem.
I want to write it.
Okay, so let's just...
I want us to realize that the chemistry of our body, the chemistry of my body is different from Dr. Aswater's chemistry of the body. The way we react to a particular nutraceutical might be different from the way she will react. And I want us to know that these nutraceuticals we are talking about are not well regulated by DFA like a pharmaceutical products. So we should be very careful. While taking nutraceutical, we should check the labeling. What are the products? What are the nutritional composition of that particular nutraceutical products that you want to take? You have to understand what is put on the labeling. Chemistry of our body. The chemistry of my body is different from Dr. Aswater's chemistry of the body. The way we react, have you tested it before? Has it been approved? Then when we check it and with the chemistry of our body, we know either we should take it or not. Thank you.
Yes, ma'am. I want to disagree with my professor. I'm not yet a professor, but
yes, it's allowed because she's talking from a phytomedicine perspective, not from a nutrition perspective.
Because you cannot ascertain or jump to that conclusion that it's not safe. Nutraceuticals, to me, they are quite mild and safe. They are weak. The supplement, they are just like our food. So that's why they say, let your food be your medicine and let the medicine be your food. So I think it addresses particular disease and infection in the body, and at the same time working for your well-being, it's just like taking carrots. Is there any negative effect when you take too much of carrots? Or it's just like taking no-cost beans? Then honey. Taking on a natural time, you would have even taken it with some things that you don't even know. It might not be that particular thing. So we should be enough time research that we be able to ascertain that is a nutraceutical causes any side effect. But for me, what I know is that it's not toxic, it's mild, so it rarely seems to induce any negative effects. I'm sorry, my prof.
I'm sorry. I didn't say they are not safe. I said genetic variations. And then the issue of honey, it's not a one-time thing. The first time I took it, I ended up in the hospital. And I thought because I took too much of it. And after 5 years, that time I was still single. After 5 years, I got married. My husband forced me and said, no, it's because you're mine. Maybe you took too much. I took it again. I ended up in the hospital. And about 3 years after, I tried it again. I ended up, I didn't take it with anything. So I have come to, I'm not saying they are, like I said, it's not that they are not safe. Just as a result of your genetic, okay, good, very good example. I'm a food scientist. There are people who do not take, there is what we call allergenicity. There are people who do not take sesame. Sesame is a proteinous seed. It is rich. However, it has some proteinous compounds that certain people react to. Then there are what we call lactose intolerance. They take milk. I'm coming. As a result of their genetic makeup, they are not able to produce synthesize the enzyme required to metabolize those compounds, and so when they take those foods, it results in adverse reactions. So it's not that it's not safe generally. So, but individual differences, like she said, individual differences in the chemistry of individuals can result to adverse effect. And that was why I said, before you take anything, please check. Be sure you have tried it and it's working for you. Okay, a very good example. I'm sorry for taking your time. There are people who do not take chloroquine. The same chloroquine has been found to be the most effective. I'm sorry for taking your time. There are people who do not take chloroquine. The same chloroquine has been found to be the most effective drug for treatment of malaria, but there are people who do not take it because they react adversely to it. And the doctor would tell you, anything you react adversely to, please do not take. Thank you.
That is
Okay, so I'd like to take us from the perspective of this question. One thing you should understand is all of us are different. That has been established. And then I'm from the perspective of this question. One thing you should understand is all of us are different. That has been established. And then I might have an underlying health factor that is not the same with you. So we can take the same things, even water, and then we react differently. Please take note of the fact that we have said your genetic makeup and then underlying health conditions that are not the same can also impact on your reaction when you take certain nutraceuticals. Now that we've established that, can we? Yes. So now that we've said that nutraceuticals are different from pharmaceuticals, from chemo therapeutics. So I'd like you to know. I'm sure this audience wants more. Okay. For those of us who are just hearing for the first time, I'm sure there are many people. Please, if you know you have heard for the first time nutraceuticals, I'd like you to know. I'm sure this audience wants more. Okay, for those of us who are just hearing for the first time, I'm sure there are many people. Please, if you know you have heard for the first time nutraceutical, that word, can you raise your hand? Nutraceutical, if you're hearing it for the first time, can you raise your hand? Thank you. At least some of them are not shy. So how do you put that word? Can you raise your hand? Nutraceutical, if you're hearing it for the first time, can you raise your hand? Thank you. At least some of them are not shy. So how do you put it out to more people out there about nutraceutical? What's the way to promote this so that people can also benefit? Because you just mentioned your research. Me that is in the university, I have not read it.
Okay, like I explained, like I said at the beginning, nutraceutical, actually the concept was propounded by, a doctor known as Dr. Stephen D. Felix. Now, he found out that certain nutrients or food components, apart from providing nutrients that the body requires, they are also able to help or confer on the body preventive measures. They prevent against certain diseases. I will give example. An example is, there is what we call dietary fiber. You find it commonly in grains. Grains are things like our cereals, wheat, sorghum, millets, and the rest of them. Now, that dietary fiber, it is important for the body. The body requires it as a form of nutrients. However, it can also help in preventing disease. One of the diseases it helps in preventing is colon cancer. How? Now, when you ingest or you take food that is high in dietary fiber, your body does not have the ability to digest it. It is insoluble. Now, when it gets into the gastrointestinal, goes into the small intestine, then the large intestine, it is able to act as a we call it prebiotic. All right? It acts as a prebiotic. That prebiotic, it does two things. Number one is that it provides food for certain microorganisms in the intestine. These microorganisms are known as probiotics. Those microorganisms are beneficial for the health. They do a lot of things, but I will not go into all of that today. But I'm particular about the prebiotic. Now, one of the things that food component does is that it helps in building bulk in your system, It helps, it provides fiber to your system. So you are not constipated and you easily pass out stools, all right? And that way, your colon, which is your large intestine, do not get stressed when you have to pass out stool. It helps in passing out bulky stool, and that helps in preventing colon cancer. That's one. There are two parts of the diet fiber. There is the soluble, there is insoluble. Now, one of those parts also help in improving the rate at which your body accumulates, or sugar, sorry, the system converts starch to sugar. And that way, it helps also in another form to control diabetics, like what she was talking about the other time. All right, that's just nutraceuticals. So, and you find this in foods that are common to us, plant products that are common to us. Many of us are so used to herbals, and that is what our traditional medicine or alternative medicine therapy is all about, because these plants, they contain what we call the components. These components have the ability to confer preventive measures of protecting the health of individuals. I think I should stop and let somebody else talk. Thank you.
Let me just add to what Professor Omogab just said. How many of us know ginger here? Ginger. Hello? You know ginger? Good. It's a typical nutraceutical. If you are having constipation and you don't know what to do, just prepare your tea. Add a 5 ml of your ginger, add to it, drink it. Before you nail it, it will flush out your system. You'll be able to go to the toilet. It means the ginger is served as a food, and same time it's served as a medicine that is useful to your body. So whatever is served as a food and medicine is nutraceutical. Is that okay? Thank you.
Thank you, ma. And we also want to add into it. When we talk of nutraceutical, what my senior colleagues have said, we generally look at it as food supplements. But I would like to advise that everyone, especially when you are above 40 years, especially for women, there are some nutrients, there are some things in your body that will not be able to come out the way it is when you are younger. So you will need some extra support, which can be in form of capsule, dietary supplements, which are these nutraceuticals. For example, these nutraceuticals work particularly to prevent or delay aging. So it really helps, especially there are this thing, you told me enough time. I'm sorry, it was there's one particular drug she told me, all women.
Okay, well,
I'm not advertising for that company, but I believe that there are some genetic disease that is in your body that you might not even know, but if you really know that your parent has something like that, you can look for a food supplement or a nutraceutical or something, or fruits that helps to reduce the effect of that thing. I actually wanted to add to that. I'll remember as I'm going. Thank you.
So a lot have been said about nutraceuticals, and they're all positive. By any means, are there any side effects or risks associated with taking nutraceuticals? Please throw some light. If you have scientific backings, please do well to tell us.
Yes. Of course, nutraceuticals have, like I said the other time, it's part of their limitations. They have side effects. Even though their side effects can be very mild, but some of them have been reported to have very deleterious side effects. One of the most common side effects of nutraceuticals is that some of them, they cause gastrointestinal disturbances. In some cases, you take some of them and the person starts stooling and all of that. And then some of them have, they cause insomnia, that's sleeplessness, restlessness. Some of them cause terrible headache, migraine. All right? And in some cases, we don't have all that time here to begin to provide evidences, research-based evidences. Some of them have been reported to damage the liver and kidney. And that is why I'm still hammering strongly on the fact that whatever you want to take, please. Now, let me say this. We have a problem in this part of the world. And that problem is that, yes, apart from abusing things, anything, somebody said one time, if you want to hide something from a black man, put it inside a book and close it. Many of us, we are not good at reading. We are very straight-jacketed. We only read what concerns us. And so you hear that somebody was having one kind of headache that resembles the one that you are experiencing. We read what concerns us. And so you hear that somebody was having one kind of headache that resembles the one that you are experiencing. And the person told you he took ginger. And then you go and buy ginger and you start gulping ginger without sitting down first of all and reading what ginger does. Now, I'm going to give a very, I react a lot to things, and so I'm very, very careful with taking things. I'm going to give a very particular example that happened recently. You know, we used to do seminar in my department and all of that. And then each time we are doing seminar, they share soft refreshments. They'll put candies in the plates, and they put bitter cola. And then each time they bring my own, I will just remove my candies and then I will push the bitter cola off. And one of my younger colleagues said, Prof. Mrs., try this thing. Now this thing is very good. Oh, it's high in saponin. It's high, and they put bitter cola. And then each time they bring my own, I will just remove my candies and then I will push the bitter cola off. And one of my younger colleagues said, Prof. Mrs., try this thing. Now this thing is very good. Oh, it's high in saponin. It's high in this. I said, I know. It's high. Another day again, I said, okay, in my life I've never taken it. Let me just try it. And I took it. And I just took bites, little 1, 2, 3 bites. I won't lie to you, for 3 days, my chests were heavy. And so the next day he told me, I said, be careful, oh. Don't recommend anything to me again. Be eating your thing. And he was like, what? Thank you very much.
Thank you very much, Prof. Not only to get the side effects, we must know the dosage of the nutraceutical we are taking in. There is need for us to know the dosage. You know, there was a time my husband was in UK and I advised him to be taking ginger, garlic, and he said,
thank God for internet. It makes things so easy for us. You don't have to go to a library and look for very big books. Just while you're lying down in your bed, go in and browse and you will get the side effects. Thank you very much.
Thank you very much, Prof. Not only to get the side effects, we must know the dosage of the nutraceutical we are taking in. There is need for us to know the dosage. You know, there was a time my husband was in UK and I advised him to be taking ginger, garlic, and he started taking it. And before you know it, the blood was going thin. Yes, he now went to the hospital. They checked him. You are healthy, but your blood is thin. He now said, I know the problem. That is garlic. Let me stop garlic. I will come back 2 weeks. And he started taking ginger. He stopped garlic. He went back, and everything was okay. So if people are telling you about particular nutrition, it is good, they are safe, but you must know the dosage that you are taking in. Before, you know, I said earlier that the chemistry of the body of Mr. A might be different from that of Mr. B. You have to understand the chemistry of your body. And one of the effects of this nutraceutical, it can be causing headache, dizziness, kidney problem, just like what she said. But let me tell us this one. I want us to take this one home. First thing you do in the morning, drink water. 2 cups of water, if possible warm water, to wash your toxin away from your body. Then take your fruits. You can take a plate of different fruits combined together. It is healthy. Thank you very much.
I want to also add to this. It seems we are trying to, because I have here in mind, kindly permit me to read this. Nutraceutical can be consumed via food as ingredient with natural high content or substances being enriched in a food matrix. At the same time, nutraceutical can be consumed as normal food. You know, at the same time, the conversion of nutraceutical through the tablets, pills, capsule. So in times of having it in food supplements, eating it as food, normal food, I don't think there should be any limitation. The food, the vegetable you are eating, when you know that, have we even eaten enough in per day? When you know that you are eating vegetable, let our food be balanced. That's what I said the other time. Let our food be our medicine, and our medicine be our food. So if we take this balanced diet regularly, we will hardly visit hospital. That's a scientist said, take an apple per day, and a apple of your meal per day. Let's watermelon. All these things looks difficult or expensive, but actually, in a true sense, they should be part of our normal food, and that is the nutraceutical me I'm agitating about. As it's different from the.
Thank you very much.
Please, audience, if you have questions, you can kindly pass them down to me. We'll read them out. We'll just take 2 questions in the session now. Please raise your hand if you have a question. Dr. Tiamiyu, you are recognized. Another question? Thank you, ma. Please come forward and ask your question. Quickly, we're rounding off the session. So one word you must remember is that nutraceuticals are part of the nutrients in your food, and they can also serve as medicine. So in all you're doing, please take moderation, pay attention to the things that you eat that you are not okay with.
Thank you very much for that lively discussion. I want to ask these questions. When you are discussing, we just want to know the difference between these nutraceuticals and the phytomedicine, herbs, nutraceuticals. These all based on plants. Now, let's just tell us the difference between the two.
Is being enriched in a food matrix. At the same time,
that is nutraceutical, is toxic, so way or that way. Please try to lay more emphasis on that area. Thank you.
Thank you. I have question and contribution. Like Dr. Mrs. was talking about phytomedicine and maybe about medicinal. Then I want to ask that some of these medicinal plants that have traditional claims, and in using them, how do laymen or layman, how do they know the level of damage that some of these medicinal plants can do to the system? Then how do we know either they are synergetic or they
visit hospital? That's a scientist said, take an apple per day, and a apple, let it be your part of your meal per day. Let's watermelon. All these things are normal food, and that is the nutraceutical me I'm agitating about. As it's different from the one
in the prevention of diseases. Dr. Tiamiyu, you are recognized. Another question? Thank you, ma. Please come forward and ask your question. Quickly, we're rounding off the session.
So how do we get them to know this? Then I want to ask the nutraceutical experts, are nutraceuticals curatives?
Okay, first of all, let me answer. Even though, because of our culture and our tradition, tends to shift or tilt towards going for traditional medication. But basically, it is not advisable for the local way they do it, like you talked about leaves and all of that. Why? Because their production process has not been standardized. Many of them combine a local, and let me also say, these nutraceuticals or herbal preparations and all of that, they interact with our system, the normal functioning of the system. They also interact with drug. There is what we call drug-to-drug interaction. They interact with drugs, other drugs that we're taking. Sometimes the interaction can be negative or positive. And so, it is advisable, if you must go for nutraceuticals, please go for the ones that have been regulated and under standardized procedures. We have them. go to the pharmaceutical. There was a time I needed, one of my babies was sick and was stooling, and I went to the pharmaceutical and I said, please, I need, and then they said Flagyl. I said, no. He now said, okay, do you have a probiotic? Probiotic are microorganisms, all right? But now they have been compounded, lyophilized and compounded into pills. And so that is a nutraceutical. Now, that's probiotic. When it's taken, it does a lot of good. So there are some that have been produced under standardized processes and that are safe. It's better to go for those ones. Okay? And there was another thing she asked about. Okay.
I think, no, I will not talk because mostly they are from phytomedicine. But let me say this. I think phytomedicine or plant extract for medicinal purpose. While nutraceutical is gotten from the food, either food, plants, or animal, from natural thing. Then let me say that there is need for you to take this nutraceutical in calculated amount. Permit me to use that word. You must know the dosage of what you are taking. Not because somebody just say you should take it, you are taking it. You must take the percentage. There is particular percentage that you need to take. Like when I produce my bread, we use 80 grams. We not just say it's bread. People are taking bread. You must have a dosage of what you are giving out. And please let us take note of this. This nutraceutical, before we start consuming it, will undergo some chemical analysis, the preclinical trials, clinical trials. You are tested in several, just like what the speaker, when the speaker was talking, how the drugs as passed through preclinical, clinical. The same thing applicable to nutraceutical. They have to pass through all these trends, and it must be labeled before you take any nutraceutical. Share the labeling, like this exotic 5-alive now. They are labeling there. The composition, know the people that are made for before you take it. Thank you very much.
Please, I will want to. My own is different. I don't know why. Because it seems I'm different because phytomedicine. When it comes to phytomedicine, what I grew to understood or what I study is that it's safe. But we have a problem in Africa. We tend to accept the foreign. The foreign language. There is nothing that is not toxic, but why do we assume? We quickly judge when it comes to medicinal plants. How do we know that is that particular plant or food? If nutraceutical is toxic, or you see that you have to take it in the quantity, then language. There is nothing that is not toxic, but why? Nutraceutical is especially for prevention, to prevent that disease. So that is why it's been advised taking it daily. But when the disease occur, the antioxidants in nutraceutical will not function as pharmaceutical anymore. So nutraceutical is to prevent that disease. For example, somebody having traces of cancer, you have symptoms of cancer. It's advisable start taking soursop. If you take soursop, all our fruits, they are benefit, they are all seasonal food, they are benefit from God. So that is for our mankind. So we should not quickly judge that why long time research have discovered that is that medicinal or the nutraceutical that have caused it. Then we should take it as our normal food and take it as supplement. As you are buying your normal food, buy your fruits with it. Then you take it as normal food supplements.
Okay, I just remembered, I want to answer one question. She talked about, are they curative? Now, the nutraceuticals are basically meant to prevent. They are preventive. They do not cure.
Or food. If nutraceutical with toxic, or you see is that
another term we use for them is that they
have to take it in the quantity. Then let's take it as normal food supplement. Take it as your food. For example, now you see that somebody that is having cancer, I always advise that person. You know, there's something between this nutraceutical and treatment, pharmaceutical. Nutraceutical is especially for prevention. To prevent, Dr. Tiami, that's what is the difference between nutraceutical, phytomedicine. You know, when we talk of herbal medicine, we are talking of using plant parts or all plants to address a particular disease. So when you are talking of what is of cancer, you have symptoms of cancer, it's advisable start taking soursop. If you take soursop, all our fruits, they are benefit, they are all seasonal fruit, they are benefit from God. So that is for our mankind. Is that medicinal or the nutraceutical that can cause it? Then we should take it as our normal food and take it as supplement.
Now the nutraceuticals are basically meant to prevent. They are preventive. They do not cure. Yes. Another term we use for them is that they manage, they help in managing.
Exactly.
Action.
Not nutraceutical
Not
anymore.
throwing away your medication and sitting down with the nutraceutical, because that's the problem. Most of us, we find that it's cheaper and we leave our medication. Please, the medications that have been prescribed, go with them, and also contact your health consultant or professional to find out if you can combine it with any other nutraceutical you are using. Thank you very much. Okay.
Sorry for taking our time. Then this question from Dr. Tiamiyu, that what is the difference between nutraceutical, phytomedicine? You know, when we talk of herbal medicine, we are talking of using plant parts or all plants to address a particular disease. So when we are talking of what dosage or something, herbals use the whole plant part. But when we are plants, so what happens now is that the way our forefathers take those plant nutrients actively into our body without isolating one or the other. But when it comes to this pharmaceutical or nutraceutical, we've been isolated for one. The active ingredient that is supposed to be synergizing it for the side effect not to come up has been dropped. So that is I will preach this way that we should take our drugs the way our herbal medicine rather, the way our forefathers have been taking it. Because as we are saying that there are side effects, we are forgetting that there are active ingredients that have been dropped from what they have been taking before. So there will be reoccurrence of side effects.
So thank you so much. That's
Let me say this, man, before you go. With some medication, take adequate balanced diet. Thank you very much.
Okay. Then your lifestyle.
Thank you so much, facilitator. They have lots to talk about. I'll just help the first person ask the question about phytomedicines and nutraceuticals. This is not about medicines, however, it's phytomedicines have to do with using roots, stems, flowers, seeds, the way they are crude as medicine, and they have different procedures through which you extract them for that use. But this is nutraceuticals. It's about your food. It's about the nutrients that you take into your body and how they can be preventive. And yes, of course, there are toxicity procedures that we do before we come up with side effects and then all of that. So if you have many more questions, you can please meet these beautiful ladies outside, or you can bring the questions to me. I'll read it to the person who can answer it, and I'm sure answers and reward event. Ladies and gentlemen, please clap for them as they go to their seats. So what was going on, a question came up for our last.
Let me say this, man, before you go. With some medication, take adequate balanced diet. Thank you very much.
Okay. Then your lifestyle.
Dosages before we come up with side effects, and then in all you said about drugs, from synthesis to metabolism, if you have many more questions, you can please meet these beautiful ladies outside, or you can bring the questions to me. I'll read it to the person who can answer it, and I'm sure maybe I think the person is trying to say, from what that you have said about drug, from synthesis to metabolism, what do you advocate, or what do you advise? We have a lot of drugs, so if we are doing precision medicine for one individual on the globe, how are we going to, what do you advise? Is that your question? Does that reflect your question? What does he advise that do about drug toxicologists in terms of drug precision, seeing that we have a vast population? How do we cover up? Is that not what you're asking? So, sir, please kindly answer.
Okay. The truth is, toxicology is a major area when we look at development of any drug. The drug might be effective, but if it is not able to selectively and treat, then it's a problem. Toxicology itself, yes, leans more on the biology angle of the scientific process. I'm sure we figured that it's a full multidisciplinary program trying to develop any drug. Okay, so the reality is we synthesize compounds, and then the level of toxicity associated with a particular compound or drug. But ultimately, the reality is that for anything to make it through the drug trial process, it must be tested, both using computational techniques in animals and then, of course, in man. So, if I understand the angle from which the question is coming, is what can we do in terms of, you know, the fact that, okay, many things are toxic, or many compounds in quote are toxic. Well, the truth is, we don't need to have 10,000 drugs, actually. If we have a few that make it through, but they are effective, then it is fine. However, for individual peculiarities, then there is need to better understand the history of the person involved, the genetic makeup as well. Now, remember that there are also developments on the biology interface. So, biologically, they can.
Maybe I think the person is trying to say, from what that you have said about drug, from synthesis to metabolism, what do you...
And look at a person's or understands a person's, the whole thing about gene editing is, I mean, they are understanding better the whole concept of the genes, the chromosomes and all that. So it's not only a chemistry matter, it actually has to be multidisciplinary. So it's to be taken from both sides, as well as, of course, clinical science itself. Taken from both sides, as well as, of course, clinical science itself, to address the issue. So, but that there are also developments on the biology interface. So, biologically, to have what we refer to as, you know, selective treatment for cases.
Thank you so much, sir. Thank you for that. I hope that answers the question. Okay, so now moving on, we're going to have our next plenary session.
Is, I mean, they are understanding better the whole concept of the genes, the chromosomes, and all that. So it's not only a
My Dean, Professor Nkiko, let me align myself, Professor Nkiko, let me align myself with the protocol, existing protocol. I'm reading K. Amodu. Molecular Biology and Public Health at the esteemed Institute of Child Health, College of Medicine, University of Ibadan, Nigeria. She's also the director of the Center for Molecular Bioscience and Medical Genomics, University of Medical Science, Ondo. Her academic journey includes the attainment of a master's and doctorate in molecular biology and genetics from the University of Ibadan, followed by postdoctoral training at Harvard University in Cambridge, USA. Throughout her career to various public health genetics research initiatives focused on childhood infectious diseases and non-communicable diseases. Our primary expertise lies in human genetics, genomics, delving into the molecular mechanisms underlying disease pathogenesis, particularly in infectious diseases and more recently, non-communicable diseases. Please let's share her up Let's give her applaud her. Her pioneering work in exploring human and parasite genetics, variability in malaria pathogenesis, has yielded groundbreaking insights, fostering collaborations with both local and international experts, including significant involvement with esteemed consortia such as the Malaria Genetics Epidemiology Network, Malaria GEN, and EU-funded networks. Her scholarly contributions are substantial, encompassing over 120 publications, comprising data sequence databases, and patents with a Google citation count of 3,142. Achieving tenure at the University of Ibadan marked a pivotal moment in her career as she dedicated herself to establishing a genetics molecular biology laboratory from scratch at the Institute of Child Health. A central goal for Professor Amadu is to enhance the capacity for research and application of genomics, bioinformatics, biotechnology, and molecular sciences in addressing infectious and non-communicable diseases of public health significance in Nigeria. Our efforts to integrate basic and clinical research into translational studies aim to elevate patent management strategies. Demonstrating our commitment to education and skill development, she spearheaded the creation of a groundbreaking academic master's and PhD program in public health biotechnology at the Institute of Child Health. This innovative curriculum intertwines essential aspects of biology, genetics, genomics, molecular biology, bioinformatics, biotechnology, epidemiology, and public health. The success of this program is evident through the graduation of numerous students across various levels, with Professor Amadu supervising several PhD, MPhil, MPH, and MSc students. Furthermore, our leadership extends to driving initiatives for technological advancement in education, notably establishing several PhD, MPhil, MPH, and MSc students. Furthermore, our leadership extends to driving initiatives for technological advancement in education, notably establishing the first hybrid electronics classroom at the University of Ibadan, in partnership with the First City Monument Bank PLC, FCMB, fostering an environment conducive to borderless learning. Since ascending to the position of professor at the Institute of Child Health, her influence has been significant, serving as acting as substantive director of the Institute at different times. Noteworthy contributions include a role as the principal investigator for the Bill and Melinda Gates Grand Challenge Exploration Grants, focused on devising innovative strategies to enhance vaccination demand among working mothers in the Ibadan metropolis. Additionally, she remains an active member of the American Society for Human Genetics, contributes as an expert reviewer for granting bodies including the EDTCP3, and holds the esteemed position of Deputy Editor-in-Chief of the Nigerian Journal of Child and Adolescent Health. Ladies and gentlemen, let's welcome Professor Amadu as she delivers a lecture, and holds the esteemed position of Deputy Editor-in-Chief of the Nigerian Journal of Child and Adolescent Health. Ladies and gentlemen, let's welcome Professor Amadu as she delivers a lecture. You're welcome, ma'am.
Can I lift it up here? Good afternoon here. Thank you very much for taking the time to listen. The title of my presentation is Genomic Insights and their application in modern healthcare. Okay, so I'll run through this. This is my outline. I'm going to run through it. We'll talk about the genome and what the application of genomics is in healthcare and what the future direction. So we're starting off with introducing what the genome is. And the genome is a complete set of genetic information that is encoded in any organism. So I have a total number of genes, and if you are going to deal with me, you talk about my own genome. So that's what a genome is. And it differs from different organisms, from different species. For the humans, we have about 3 billion pairs of DNA, and they are organized into 23 pairs of chromosomes. From our basic biology, we know we have 22 pairs of autosomes, and we have 1 pair of sex chromosomes. For the female, you have the XX, and for the male, you have the XY. So that is simple biology. We are going back to simple biology. And all genes are important for development, for functioning, and reproduction. And interestingly, we are still doing a lot of research to know the function of some genes. So with the Human Genome Project, which I'll still talk about later, we've been able to discover a lot of genes, and scientists have been able to, they're still continuing the work to understand the function of some certain genes. Now, we need to understand the human genome. This is crucial, because when we understand our genetic traits and how it affects our diseases, and it will help us to be able to know how to, drive healthcare. The previous speaker, though I didn't stay around much, talked about using synthetic, developing drugs. And now we are talking about there's a site developing drugs, and you cannot develop drugs in isolation or silos. We must now begin to think of how the ones developing drugs, we also work with people who are looking at the genetic traits. Talked about using synthetic, developing drugs. And now we are talking about there's a site developing drugs, and you cannot develop drugs in isolation or silos. We must now begin to think of how the ones developing drugs, we also work with people who are looking at the genetic traits, so that we will see what are the important things. As we go on, I will explain a little better why this is important. Okay, so genomics, I'm sure you've heard about omics. There's so many omics all over the place, but genomics was the first omics. And because of the rise in innovations, development in molecular biology, it started a drive in the 1990s. I will go through that developmental stage. And that was when the Human Genome Project started. It was about 1990. And they started the work, little by little. Eventually, they were able to get the work done by up to a point at 2004. And I can tell you that since then, we now call that the post-genomic era, meaning that after we discovered the human genome, the era coming behind it, there has been a lot of discovery since then. It's a very interesting time for us if we'll just key into it. So genomics is the study of the entirety of the organism's genome, how we interrogate the different variations in our genes, and how these genes contribute to what we call Mendelian diseases and other complex disorders. So I'm giving an example of the other omics. I'm just going to mention them in case you come across them, so that you don't just say, which one is this omics again? You understand what they are all about. So we have proteomics. That is the study of proteins. Then we have the metabolites, metabolomics. So metabolites are also important in this era. Then we have the environmental DNA, which is metagenomics. That means from the environment, you can track diseases. That happened a lot during COVID. In many, from maybe the African American or Africa or Europe. So we need to be able to classify and categorize our phenotypes. So that is why phenome-omics is very important now. So what are the important developments in genomics? This slide is busy, and I'm not sure how we are going to be able to look at it, but if you can see that it started from 1990, just as I mentioned. That was when the Human Genome Project started. And there have been a lot of challenges along the path. Eventually in 2003, they completed, I would say at that time, what they knew about the Human Genome Project. And then afterwards, we had something called the first HapMap report. In fact, they came to Nigeria and they used the Yoruba population. And that project is so important today. Why is it important? Now, when we see, when we type different genes, we're able to compare it with this HapMap project and see, what are the minor allelic frequencies? Why are those things necessary? Those things are necessary because, for example, if you have a particular gene that may be associated with a disease in a population, by doing the genotyping, you'll be able to know whether that gene is actually prevalent in our population, maybe to a large degree, and then you'll be able to see which one is actually the at-risk genotype. For example, we all know about the HBAA, HBAS. Please, we all know that the S is the risk one, right? That is actually the phenotype. We know that the S is the risk. So that is why we need to do all these projects. So in 2005, we had the first HapMap. Then we had 1,000 genome projects. And then in 2013, there's a gene editing tool that was discovered. I will still talk about that a little more, but I'm not going to bore you with too many technical details. So by 2022, the entirety of the human genome sequence was completed. So even though a part was completed in 2013, the full thing was now completed in 2022. So this HapMap, like I mentioned, they were landmark projects, and they help us to understand more about our genes and how they vary from one person to the other, or how should I put it, how the genes affect diseases. How, if you have a certain variation in a gene, it may make you more susceptible to a disease or not. So this is just the example of the HapMap project, the 1,000 genome. So from there, it's a catalog of what you call single nucleotide polymorphisms. These are different variations in our genes. So with the HapMap project, there's a haplotype map, and it has common patterns for human genetic variation, and it's a very useful tool for those working in that field. And then on top of it, we now have the 1,000 genome project that identified about 84 million SNPs and different variations in the human genome. So what are the tools for what are the genomic tools and technology that we have been able to see in these last few years? Again, there's a map showing that a study for genomics even started in the 17th century. There's a report of somebody observing an oral microbiota, even from back then. But then study started slowly, little by little, and it seems as if in the 20th, 21st century, everything just boom, and there's more knowledge available. Now we have a lot of studies going on. We can talk about so many genomics, and these genomics these omics come about because we were able to develop many tools. For example, we just had the Sanger sequencing to start with. Then afterwards, biotechnology developed, and people began to develop different sequencers, and we now have varieties of sequencers that we can use. So now we are sitting in a time where we can answer our problems, and I just want to challenge us here in Nigeria, there's still a lot of work to be done. We can sit down, I can do the entirety of this presentation and talk about what people have done, but I will keep interjecting on the things that I think we should start paying attention to, because that is important. I'm not just here to just give a lecture and that are happening in the genomics world, and we can do that and close shop and go home, but I think we can start, thinking about the little things we can start doing to start building. They started from somewhere after all, and we should not be in a hurry. We should start off from somewhere so that we have extreme data that is useful to us here in Nigeria. All right, so we have these different genomic tools. We have the PCR. It's expensive, but I think I've given a talk here before that I mentioned that, yes, these things are expensive, but we can start, little by little. We can combine with each other and start, answering some questions little by little. So there's the genomic, so we are here. There are other tools available. There are SNP analysis, genotype arrays, and by collaborating with people, we can start using these tools for ourselves. So we can do a lot of things, even with the little that we have. And I just want to encourage us that we shouldn't just think that it's impossible. Nobody is going to type our population the way we can type it. All right, so we have next generation sequencing now, and that is high throughput. We can do a lot of whole genome sequencing. We can do genome-wide association studies. We can partner with different collaborators. The interesting thing about combining with other partners is that, yes, they use your data for the large pool, but you can retrieve your data and use it to apply it to your own population. I was looking at some of the data I'd collaborated with a large consortia with, and I realized that there was a paper that I did we're doing phenotyping studies, and we're looking at how sickle cell traits, that is AS, and ABO blood group is associated with severe malaria. And our findings show that children that have group O blood group, they protect against severe malaria. But children with blood B group, are more susceptible to severe malaria. And then I now saw that in the general large data, they actually confirmed it with their genome-wide association studies. So now I can authoritatively say that children, if you have a child that has B blood group, that child should be watched even more carefully than other children, because that child can come down more with malaria. So, those are ways we can answer our own problems and, share our data here. Okay, so those are ways we can answer our own problems and, share our data here. Okay, so there are other tools, the CRISPR-Cas9, and then the bioinformatic tools. I want us to encourage young people around who are into computer studies. We can do a lot with bioinformatics, but we don't have as many people who are interested in bioinformatics, and we need that particular field to be able to answer so many questions. So I'm throwing a challenge to even the younger people. Get involved. You can play a role. All right, so what are the applications of genomics in modern healthcare? We've heard about precision science, precision medicine. Disease prediction is one of it, and prevention. Pharmacogenomics, genomic testing and screening, gene therapy. How do we manage infectious diseases and reproductive health? So and they're doing a lot of this even with reproductive health. We have many clinics now that do, I've forgotten, what's the name? They type the mother, they do a lot of genomic testing now before, when, please remind me, ma, a woman wants to get pregnant. In vitro fertilization, IVF. So, yes, IVF. So there's a lot of testings that are being done now, especially in high-brow ones, maybe in Lagos. They will do a lot of testing. So some women can even ask for designer babies if they want. I'm just joking. But, there's a lot of genomic testing going on, even in these places. So please let's just sit down in the university, close our eyes and think of just promotion. Please, let's think beyond that. There's a lot we can contribute to our own healthcare. By the little studies that we do, they add up. You understand? You do your own study, you give a result. That result is going to be useful to somebody else who is also doing something else. So that is the challenge I'm throwing to us. We all can play a role in this genomics in modern healthcare. All right, so I see that there has been a group before that talked about using Nutri-Sequel for medicine. So there's evolution of medicine. Initially we had the traditional medicine. There are the long established practices and treatments. Then we now have evidence-based medicine. That means that it relies on scientific evidence for you to be able to make clinical decisions. Now we are going to precision medicine, where your genome is going to help the clinician to decide what kind of treatment he's going to give. And I think we need to challenge ourselves to get into that bandwagon. It's very important. One size does not fit all. One size doesn't fit all. I remember even simple malaria drugs. It works for some people. It doesn't work for some people. You know? But, so you see somebody who takes one drug and says he has malaria resistance, he has parasite-resistant malaria. It may necessarily not be the parasite that is resistant. It may be that he himself is not metabolizing the drugs well. So there are things we have to start thinking about now. You understand? So we have people like that. They may take 3 or 4 different drugs before they feel better. So it's not one size that fits everybody anymore. So we have to start thinking of this precision medicine. What is it really about? It's an approach about disease treatment and prevention that considers your individual variation in your genes, in your environment, in lifestyle. And now they are even categorizing it into personalized medicine, stratified according to a group, tailored medicine, maybe according to your, So then P4, predictive, they predict your risk. They see how can we prevent it. They personalize it. Then you are even participating in your own treatment yourself. So there's a lot going on in the world about precision medicine. So this slide, I want to show in this slide that you say a large group of people and you are giving to them. In that group, some people in red that have serious side effects. How many people have seen that before with Fansidar? Have you ever seen the side effect of Fansidar before? I saw one time, it was terrible. It's as if the person had burns on his body. And that's because of the sulfur in the Fansidar. So, for different people, and that's about 1 in 100. You understand? So, some people will have serious side effects. Some people will have no response. And maybe majority will have desired response. And we will say the drug is working. For those other categories of people, it's not working, right? So we need to begin to think about what the genetic variation is. So we now know something that is called pharmacogenomics. So we have pharmacology on one side, the effectiveness of the drug metabolism, the side effects, and then on the other side, you have genes that have been inherited, you have the environment, what we now consider as pharmacogenomics. So with pharmacogenomics, we need to understand differences in the genes and how the body responds and the body's response to these medications. Does that person metabolize the drug or not? It's a combination of pharmacology and genomics to be able to understand genetic variations. And that's why Professor Lomola said that in answering the question they gave to him, he can't just answer by himself. There's also the genetics aspect that we have to consider. So even if we develop drugs, we now have to start thinking of not just only the side effects. We now have to start thinking about the genetic variants. Will this person with this kind of genetic makeup be able to metabolize this drug well or not? So there's a lot to consider. And that's why we are all important in this, conversation. Everybody has one little thing to contribute into this conversation. So what is genomics' role in precision medicine? Genomics, is central to identifying the genetic mutations that impact whether you are at risk of a disease or you can respond well to a treatment. So you have the gene specific, you have the genomic-driven diagnosis. So those are the things, that is how we contribute to precision medicine. And then you can use biomarkers. There are different types of biomarkers to track the disease or whatever. So those are the useful things that genomics, that's how genomics is very important in precision medicine. And then you'll be able to clear diagnosis, you'll be able to say, oh, this drug targets this particular biomarker, for example, and will be a useful treatment for this particular disease. So these are the different biomarkers that we have. You can have a biomarker that monitors disease. And biomarkers can be proteins. So once you identify that this particular biomarker is associated with a disease, then it can be useful. It can either be monitoring, it can be a predictive biomarker, it can predict how severe the disease is. So those are the little studies we can do, basic science, clinical people combining together. Let us see particular protein, is it associated with a disease or not? Or can we use this marker for diagnosis? Those are the different types of biomarkers. So, so in genomics, we now have many useful studies. We have GWAS, that's the Genome-Wide Association Study. Like I gave an example of how a Genome Association study confirmed that, B blood group is susceptible to severe malaria. You have millions of markers, and then when you find these different markers, you can then take them in your population and then do a study to say, okay, they found that all over the world that this particular marker can track a particular disease. Fine, they've published their own results. But the truth is that biomarker may not be useful in our own population. So we need to answer that question ourselves. Maybe it's a different marker that is useful. We need to be able to understand. Such studies where a particular gene that was found in Europe to have different variations, and in the population it doesn't for our own population. So we have whole genome sequencing now. There's a whole genome, maybe concerning, you have cases, you have control, you'll be able to see what particular variants are more associated with the disease. And then you have whole exome sequencing, where it will sequence all the protein coding parts of your gene. And then we now have single genomics now. Now, with this one, it focuses on the genome of individual cells. You know that our cells also have different genomes. And sometimes there's something called mosaicism, where there'll be partial inheritance. There's some funny thing in one particular cell, it may be different for the next cell. Okay, I can use this one. So, single cell genomics is now even more specific. So there's a lot to get into. And I keep telling people that we can sit and complain that we don't have money and all that. But fine, let's start from the basics. The basics are so important. And I'm glad I'm talking to Faculty of Science. You understand? We are so relevant in this field. It's so, medicine without science is not going anywhere in these current times. That's the truth. There's a lot to do together. The doctor in the clinic will get frustrated if all he's doing is prescribing drugs that are not working. My own PhD started with a doctor like that. They've been seeing children with severe malaria in the clinic. And he said, why is it that one child will develop cerebral malaria in a family and the others will not? What is the cause? And there are many reasons why. There's the child's genetic makeup, there's the immune response, you understand? There's even the parasites virulence as well. So there are so many things we need that we can do, that we can start from the basics. Each time I'm talking about these highfalutin tools, I keep coming back to the basics. Because when we look at the highfalutin tools, we become discouraged. But that's why I keep coming back to the basic ones, because we need that basic. It's like building a castle. Without a foundation, there's nothing. So I'll keep going on. So genomics is so important for infectious diseases, for pathogen diversification, to know the different infectious diseases. We can track infectious diseases in our population. We can track when we have large, like we had COVID, well, we don't even, I mean, we can have different outbreaks, and we can type the last outbreak with the current outbreak. What has changed? What is different? How can we prepare for the next one? If it mutates, we can predict that it may mutate this way, so we'll be expecting the next outbreak. So there are many things to do. With HIV, tuberculosis, there's a lot of work that has been done on malaria. See the huge amount of details we have from COVID-19 just because of these tools that we have. So these are things, there are even different projects now, various platforms that support pathogen identification, outbreak tracing. There's something called IDSeq, Next Strain. For those that work particularly in infectious diseases, there's a lot of tools available. There are many projects that we can write and, you know, present our data. Our data is so important, even in the global space. So those are things we can do. There's an example of a center, the ACE-G, the African Center of Excellence. Started from somewhere. Today, they played a key role in the Ebola, even in the COVID-19. And even with a lot of collaboration, they were able to have rapid diagnostic tests that were developed. Even in NIMA, they developed a rapid diagnostic kit for COVID. So there are so many things we can do. We can work with people that have experience, we can learn from them, and we can do the little that we have. Our data is so important. Even we can get variations from different, you know, many of these networks have typed these variations, and they are looking for people who will come and do analysis on them. For example, Malaria Gen has a lot of variants on P. falciparum, and we can use this data. We can start thinking about how to develop vaccines. Do you know that during the COVID-19, it became so very obvious that we don't have any vaccine-producing centers in Africa, even till now. Even till now. So we need to get into this discussion, get into this conversation. I'm just going to run through these cancer treatment options. There's a lot that has happened or is happening right now with genomics. For example, I'm giving an example. This particular cancer, non-small cell lung cancer, they identified what they called an epidermal growth factor receptor. That there are two important biomarkers that they found that were associated with this cancer. And then they discovered that these receptors, they bind ligands like EGF and cause a lot of dimerization and activate pathways for cellular growth. And they found another one, that is the anaplastic lymphoma kinase. These are two important markers in this lung cancer. Now, they found that the EGFR has some particular mutations, and discovered that these mutations can be targeted by an inhibitor. So it is that inhibitor that they now took out. And, you know, molecular targeted therapy, that's what they call it. So it's like finding an active ingredient in a plant. You know, you will take out the active ingredient, you will process it so that you can use it to manage the disease. So this inhibitor is from humans. So they now found a way to synthesize it, and they are now using it as targeted molecular therapy for this particular cancer. Now, can I shock you with something? That even though this works, it won't work for us in Nigeria. For example, for prostate cancer, these are a list of genes that have been associated, that they've typified and found that, you know, you can use it either for PCA screening, prostate cancer screening, and rapid diagnostic kits and things like that. In Nigeria here, when my student presented this as his MPhil, the internal examiner was so excited because they found out that the PSA levels that they are now using is not. There are many things that are going on. They even use, from these data that they have, they can use it to predict risk scores. After predicting risk scores, they will now see a particular individual that if they put all this together, all this is genes, is phenotype, and everything, they now put it together. It's like an algorithm, trying to find out that, okay, can we predict this person having this cancer, and how long will it take for found out that the PSA levels were not working anymore? So there's a lot to just challenge us this afternoon. I don't want to go into all this. We're in the afternoon. I don't want to go into too much technical details because we all get bored. That's why I keep interjecting the challenges I am throwing to us. So, for example, this person, there was this polygenic risk scores for coronary artery disease. They used what they call, it's a genome-wide polygenic score to identify individuals that had a higher risk of this coronary heart disease. Too much technical details because we all get bored. That's why I keep interjecting the challenges I am throwing to us. So, for example, this person, there was this polygenic risk scores for coronary artery disease. They used what they call, it's a genome-wide polygenic score to identify individuals that had a higher risk of this, you understand? So how can we use all the results we have from different studies? Nigerians have contributed to many GWAS studies. How do we pull all those GWAS studies, you understand, together from different sources and then look at our own population and be able to type what are the polygenic risk scores with all this data that we have from our own population in different studies. And from this person's study, they were able to discover that there were some individuals that were at greater risk, you understand, using this particular algorithm. So there's a lot for us to do, whether it's basic laboratory science, whether it's wet lab, as we call it, or dry lab, which is bioinformatics. There's a lot to still do. That is my challenge this afternoon. I don't know if population in different studies. And from this person's study, they were able to discover that clustered regularly interspaced short palindromic repeats. In fact, that's a long thing, so that's why they call it CRISPR-Cas9. So it's used for genome editing. In simple language, it cuts the DNA. And Nigeria has a large burden of sickle cell. I want to ask a question here. Are people really aware of, and then take again, of getting married to, you understand, and saying love is all that matters? So my question is, are we really doing that awareness? Because it is critical. Anyway, what I'm doing here is different. So there's a cut, they would have interjected the repair, they cut the bad, let's put it that way, the bad SNPs, and interjected the correct one in, and then there's an enzyme that will now repair the DNA. That's CRISPR-Cas9. And it's been used for cancer, and it's being used for sickle cell anemia now. And then transplant it back into the patient, and then increase the production of hemoglobin F. So with that, it reduces the sickling, and then this person can live a normal life. But again, this kind of therapy is very expensive. At least for now, it is still very expensive. So there's still research that we can still continue to do to make a difference. So what are the challenges for implementing genomics in healthcare? We just mentioned the first one: cost, accessibility. Data management is the second one. And that is why I have been saying that repeatedly. We need people who will develop interest in data management, managing vast amounts of genomic data. We need that expertise in our own population. And how do we now integrate this genomic data into our electronic health records? So there has to be a talk between the basic and the clinical. We cannot work in different places if we want to talk about healthcare in our population. So we need to find a way where we will work together. We generate the genomic data, we interpret the genomic data to the clinician. You understand? So we need to understand how we are going to work together doing this. And then there are other ethical issues. We have to talk about religion too as well, legal and then social issues, cultural issues as well. If these things are presented to people, how would they take it? Would they accept it? So those are the issues we have to talk about in implementing genomics in healthcare. Informed consent. I call it understood consent. How many times do people really? So we need to do a lot of that, informed consent, so that people understand what they are doing. And I believe that the layman is intelligent. If he understands what you are talking about and he knows the future impact that can be even to his own family, so we need to work with that, informed consent. And then if we have all this evidence, or if we do not have the evidence, so there are two ways. If we have the evidence and we don't have the connection with the clinic, how is it going to work? If we do not have the evidence, how will the clinician be able to help us? So those are the two major things in terms of challenges of genomics. So, one, we have to get the data. Two, we have to work together. So those are the major things that I see as issues. So future direction, we need to have a comprehensive training for all of us, putting together basic genetics, genomics, molecular biology, and clinical applications. Like I said before, we cannot build a castle without building a foundation. We need the basics before we can move anywhere. Then we need to start thinking about genetic counseling. This is so important. And as a medical school here, it's something we need to start thinking about, genetic counseling. And genetic counseling is not just the clinician, it involves the geneticist, it involves everybody, in every sphere of life. Those are the important things we have to think about. Then we need to emphasize the importance of ethical, legal, and social issues that relate to genomic medicine. We need to understand data interpretation skills, encourage health informatics. Again, I keep saying it. We need health informatics, we need information technology training, we need bioinformatics. These are the things we need. And we need to start introducing genomic medicine in every curriculum. Let's become aware. The biophysicist has a role to play. The chemists have a role to play. So how do we infuse all these little things in our curriculum? No, even if it's just a course in the whole year, in the 4 years, at least the student has an understanding. That I can also play a role in genomics and healthcare. This is my recommendation of future direction. So, genomics is so important. The insights will revolutionize our healthcare. And we need to be awake in Nigeria. We need to collaborate with one another. Science, we cannot stay in one place. We need to move together with the clinicians. We need to be able to talk to each other, because at the end of the day, we need to be able to talk to each other, because at the end of the day, it's our collective responsibility as a population. Thank you very much for my presentation.
deliver the lecture. Before we move on, do we have one or two reactions? Two reactions from the house? Maybe there are things you want to clarify. Okay. Okay. One of the graduate students come here now.
Good afternoon. Thank you, Prof, for that lecture. I just want to comment on the area of data management and bioinformatics. For somebody like me in computer science and statistics, we are so much interested in data mining, and then if we look at what artificial intelligence has provided for us today, it has given us opportunity to bridge the gap and the challenges we are facing formally in our attempt to analyze some of these genetic diseases. But looking at our on the fact that we are limited in terms of data that domicile in our population that we can use to model genetic diseases, that is completely missing in our own population. But we have several of them online for other population. Then our own Center for Genomics and Molecular Biology is doing to make some of these data available so that we can apply some of these artificial intelligence that are available today to address that area. That's number one. Number two, is there anything that the center can do in order to enrich our own population, the AI techniques and those bioinformatics aspects of computational biology? Thank you, ma'am. We have several of them online for other population. Then my question is, what is our own Center for Molecular Biology is doing?
And the main aim of that is to be able to collect samples, clinical data also collected. And if we have this in our bank, more or less like a bank,
that we can apply some of these artificial intelligence that are available today to address that area. That's number one. Number two, is there anything that the center can do in order to enrich our skill so that we can actually prepare ourselves and then our techniques and those bioinformatics?
It's a long stretch. But one of the ideas we have is to have what we call a biobank. Somebody has an idea tomorrow and wants to work. He doesn't need to go back to in our bank, more or less like a bank. Somebody has an idea tomorrow and wants to work. He doesn't need to go back to collect that data because data is already available. And all he needs to do is maybe he has a grant, pay a little fee, collect the samples that he wants to work with, and generate data on it. So those are one of the things that can retrieve those data from the different places where they are and pull it together. Let's have a picture of what we have in Nigeria to start with. Now, when I'm glad you are interested in something like that. Again, one of the things that we are putting down as a working strategy for the center is to have short-term courses. So short-term courses where you can, at your own pace, you can take the lecture so that you have a better idea about genomics, all the idea of what you are looking for when you are now using all data mining and doing that together.
Thank you, ma'am. One more. If there is none, I think we should proceed. Now it's time for lunch. I know many of us are saying, thank God. It's time for lunch. But before we go to lunch, before we go for lunch, please let's listen to this special announcement. If you are presenting, if you are having oral presentation today, just go through the book of abstract. You will see 24th. See whether your presentation has been scheduled for 24th. So while we go out for lunching, you will see Dr. Adibayo. You can submit your presentation to him. And for those of us that have a poster, during lunch, please just go to the back of this hall. You will see we have already allocated what is it called? Number to your poster. Just identify your number and fix your poster there. So the number is already in the book of abstract. Check the title of your poster and go there and fix it to the corresponding number. So this is the schedule of our presentation. Food science will receive a large number of entry from food science. So you may, I think it will, please let's clap for food science. I think food science is taking over Faculty of Science now. Okay, so food science will be having.
Can at your own pace, you can take the lecture so that you have a better idea about science. You have an idea of what you are looking for when you are now using all data mining and doing all these other things. So that is one of the things we also, if you want to take a diploma out of it, that will also be available.
Proceed. Now it's time for lunch. I know many of us are saying, thank God, it's time for lunch. But before we go to lunch, please let's listen to this special announcement. If you are presenting, if you are having oral presentation today, just go through the book of abstract. You will see 24th. See whether you have 4 lectures room here after here. So lecture room 2 is just, I think the one just opposite. You will see it there. Lecture room 2 to the back of this number. Check the title of your poster and go there and fix it to the corresponding number. So this is the schedule of our presentation. Food science. After lunch, just go straight to lecture room 2. Physics, chemistry, mathematics, and computer science. We have their oral presentation in lecture room 3. So immediately after the lunch, just proceed there. And for food science, Dr. Janet Igbaro. So the 3 of you will be in room 3. Microbiology and SLT will be in lecture room 4. Microbiology and SLT will be in lecture room 4. Is there projector in lecture room 4? I will confirm. There is projector there. This is a lecture 4 or 1? 4. Okay. So lecture room 4, microbiology and SLT. Biosciences and biotechnology will be here. So please let's go out for lunch. Professor Amodu for that brilliant presentation. Our dean, thank you for being patient. Our former dean, we thank you for your patience. Let me answer. Okay, that's my card. Please. Okay. We have Dr. Ogundele and Dr. Orodepo and Dr. Rotiola Famutimi. The three of you will be in room 3.